The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hello children, how do you do? Hope you are healthy and happy. Our greatest glory is not in never falling but always rising when we fall. Dear children, failure and success is a part of our life. There are people who give up after failing and they will never get up. Dear children, they must get up and they must rise up with double the energy, as never forget that failure is the pillar of success. My dear children, we can say that life is a great teacher. We should learn from it and we should be a real winner. By saying this, I move on to the lesson number 6, cell cycle and cell division. Related to this chapter, we already understood in our previous class. This chapter is related with mitosis and meiosis. So, we are just going to rebrush about this chapter. My dear children, first one is the cell cycle, the sequence of events by which the cell duplicate its genome. Later on, it synthesizes the other constituent of the cell and eventually it divides into two daughter cells. This is called cell cycle. So, what is cell cycle? It is a sequence of events by which the cell duplicates its genome, synthesizes the other constituent of the cell and later it divides into two daughter cells. This is called cell cycle. And the duration of cell cycle in organism is man 24 hours, yeast 90 minutes, bacteria 20 minutes, onion root tip 20 hours and in case of epithelial cell it takes 8 to 10 minutes. Next one, we can see the phases of cell cycle. The first growth phase is the G1 phase, then comes the synthesis phase which is called S phase, then comes the second growth phase which is called G2 phase and then comes the M phase which is known as mitotic phase. So, this complete phase is the cell cycle of an organism. In the cell cycle of an organism, G1 phase, S phase, G2 phase and mitotic phase is taking place. G1 phase is the growth and normal metabolic role taking place there. In the case of S phase, DNA replication is taking place, therefore it is known as synthetic phase, and in G2 phase growth and preparation for mitosis taking place, therefore it is called second growth phase. G1, S and G2 phase together is known as interphase. I think you would have understood about this. We can see here phases of cell cycle, 4 distinct phases are there: G1 phase, S phase, G2 phase and M phase. In the case of G1 phase, as I told you, it is the growth phase or first growth phase which is also known as starting phase, and it is a gap between mitosis of preceding round of cell cycle and DNA synthesis of current cycle. In this phase, cell increases in size, growth phase of RNA is taking place there and each chromosome with single molecule of DNA is seen in this phase. S phase is the phase where DNA synthesis and it replicates and the amount of DNA is double there and each chromosome will be having 2 sister chromatids. In case of G2 phase, it is the gap between DNA synthesis and the division phase and it is the checkpoint control before mitosis. that is related to spindle fiber formation. These three phases are related with interphase, and the fourth phase is M phase, which is also known as mitosis phase or cell division, and here the formation of diploid cells are formed after cytokinesis. So, that is related to the phases of cell cycle. Here you can see G1 plus S plus G2 phases is called interphase, which is taking 12 to 24 hours in a mammal cell. In the case of M phase, it is taking 1 to 2 hours. So, this is the representation of phases of cycle, where you can see M mitotic phase, G1 phase, S phase and G2 phase. These three together is known as interphase, and here you can see one G0 phase is also there. So, we can now see what is G0 phase. G0 or quiescent stage. It is a stage in cell cycle where cell do not divide further. So, in this stage the cell is not dividing further. It exits G1 phase and enters into the inactive stage of cell cycle. So, it will be coming out of the G1 phase and it will be moving into the inactive stage of cell cycle, and during this stage the cells will be metabolically active. Even though it is entering into the inactive stage, still the cell is metabolically active. Now, in the case of animals, mitotic cell division is taking place in diploid somatic cells, whereas in the case of plant cells, it is taking place in haploid as well as diploid cells. Next one we can see, mitosis is the division of cell in such a manner that each of the daughter cells receive exactly the same number and the same kind of chromosome that the parent cell is having. So, mitosis is a division where the daughter cell will be having exactly the same number as well as same kind of chromosome which is already present in the parent cell. Therefore, it is also called equational division, and it takes place in the somatic cell, and it was first observed by Walter Fleming. Next one is related to the stages of mitosis. Prophase, metaphase, anaphase, telophase, and later cytokinesis will be taking place. In the case of prophase, chromosome condenses and become visible. Spindle fibers then enlarges from the centrosome. Nuclear envelopes break down. Centrosomes move towards the opposite pole. That is related to the prophase. Metaphase, you can see the figure. Chromosomes are lined up at metaphase plate, and each sister chromatid is attached to the spindle fiber originating from opposite poles. Anaphase, centromeres split into two sister chromatids, they are pulled towards the opposite pole and the spindle fibers begin to elongate the cell. Telophase, here you can see chromosomes arrives at opposite poles and begins to decondense. Nuclear envelope will be surrounding each sets of chromosome, and the mitotic spindles, they are breaking down. You can see the spindles, they are breaking down, isn't it? Later on, cytokinesis is taking place. It is taking place in different way in animal cell as well as plant cell. In the case of animal cell, we can say a furrow is formed there which will be deepening into the cytoplasm and thus making the cytoplasm into two equal halves, thus two daughter cells are formed there. In the case of plant cell, cell plate is formed there, and because of the formation of cell plate, the cytoplasm is being dividing into two daughter cells. So, we can see one by one related to the stages of mitosis. Prophase, nucleolus disappear, chromatin condenses into chromosome and formation of the mitotic spindle is taking place. This is the peculiarity of prophase. In the case of metaphase, it is the second stage, and the important part of this stage is that spindle fibers are formed from the centriole and the chromosomes, they will be aligning themselves on the metaphase plate. That is the peculiarity of metaphase. Next one is anaphase. You can see the figure here again. The chromosomes, they will be breaking at the centromere, and the sister chromatids, they will be moving to the opposite poles of the cell. Next one is telophase. You can see here what is happening there. The cytoplasm is dividing here and the chromosome is again uncoiling to chromatin. Nucleolus is reappearing and spindle fiber is disappearing. Nuclear envelope is forming there, and with that two identical daughter cells are forming. Then after telophase, cytokinesis is a stage taking place there where cytoplasm is dividing. As I told you, in case of animal cell, a furrow is taking place. Because of that, two daughter cells are formed there. In the case of plant cells, a plate is formed there, and because of that formation of plate, two daughter cells are formed there. Now we can see the significance of mitosis: growth, reproduction, maintaining and repairing. In the case of growth, increases number of cells, contribute to the growth of tissues, and in the case of reproduction, we can see there it helps in some organism to reproduce asexually, and maintaining the same number of chromosome as the parent is having, and for the repairing, it repairs worn out tissues which is been there in the cell. So these are the significance or importance of mitosis. I think you would have understood about mitosis. Now next one is meiosis. It is a type of cell division in which one parent cell divide to form four daughter cells. Meiosis is a type of cell where from a parent cell four daughter cells are formed there, and each daughter cell will be having half the number of chromosome when compared to the parent cell, and meiosis is taking place in germ cell, or we can say in reproductive cell, whereas mitosis is taking place in the somatic cells. And meiosis is helping in the formation of haploid gametes. In the case of meiosis, we can say the cells will be getting haploid gametes. It was first discovered or observed by Strasburger in 1888, and the important features of meiosis are: it involves two sequential cycles, that is meiosis 1 and meiosis 2. Meiosis 1 is initiated after parental chromosome have replicated and the identical sister chromosomes are formed. And next one, it involves pairing of homologous chromosomes, and thus recombination of these chromosomes are taking place, and because of that four haploid cells are formed at the end of meiosis 2. So this is the key features of meiosis 2. We can see the phases or stages of meiosis 2. In the case of meiosis, that is the key features of meiosis we have seen just now. Now we are going to understand about the stages of meiosis. In the stages of meiosis, we can see there it is classified into two stages, meiosis 1 and meiosis 2. In case of meiosis 1, we can see four stages are there: prophase 1, metaphase 1, anaphase 1 and telophase 1. In the case of meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. So these are the stages related to meiosis 1 and meiosis 2, which are the phases of meiosis. So you can see the figures related to the stages. Now we are going to study deeply related to the stages. Meiosis 1, you can see meiosis 1 is also known as reduction division, because during this division the chromosome number is reduced to half. So we can say that meiosis 1 and meiosis 2 are the phases of meiosis, but meiosis 1 is known as reduction division because here chromosome number is reduced to half. In case of meiosis 1, number 1 is the prophase 1, it is the longest phase, and this phase is divided into 5 sub-stages. Number 1, leptotin, zygotin, pachytin, diplotin and dikinase. So 5 stages are there: leptotin, zygotin, pachytin, diplotin and dikinase. So that is related to prophase. So in number 1 we can see leptotin. It is the first stage of prophase 1 and it is also known as leptonema, where it means thin thread. During this stage the chromosomes begin to condense to form long strands, and here as the chromosome is condensing to becoming a long strand, or we can say a rod like structure, still the two sister chromatids, they are tightly bounded to each other and they are indistinguishable or they are not visible from each other. Number 2 is zygote stage, which is also known as zygonema or York thread stage. Here the chromosomes again becoming shorter and thicker. And number 2, the pairing of homologous chromosome is taking place, and this pairing of homologous chromosome is known as synapsis. And a pair of homologous chromosome which is formed there, they will be lying together, and that is called bivalent. And a complex is formed there between the homologous chromosome, and that is called synaptonymal complex. And what is synaptonymal complex? The two homologous chromosomes, they are brought together through a characteristic ladder-like structure. Ladder-like structure is formed there when the two homologous chromosomes, when they are brought together, that is called synaptonymal complex. So, that is the features of zygote stage. Next one is packet stage, which is also known as thick threaded stage. Here again condensation of chromosome is taking place there, and each chromosome is visible where two sister chromatids is seen there by joining at the centromere, and now these two sister chromatids, they are called dyad, and each bivalent has two dyads. Each bivalent will be having two dyads, therefore it is called tetrad, that is 4 chromatids are present there. Okay. Next one you can see synaptonymal complex, isn't it? That is the paired duplicated homologous chromosome assemble into an elaborated structure, that is synaptonymal complex after the pairing. And sometimes what happen, exchange of chromosome is taking place or exchange of gene is taking place, and that is called crossing over, and this is taking place at a peculiar or particular point, and that is called recombination nodule. That is called recombination nodule, which is very, very important. I think you would have understood about packet stage. So, in the packet stage, what is happening there? Recombination nodules are formed there, and these nodules are formed after the formation of synapses and the formation of synaptonymal complex. My dear children, next one is diplotin stage, which is also known as double threaded stage. In this stage, dissolution of synaptonymal complex is taking place. That is, this stage is the longest duration. Here nuclear membrane and nucleoli starts disappearing. Desynapsis or separation of homologous chromosome begins due to the dissolution of the synaptonymal complex. The synaptonymal complex which is formed there, it starts to separate from each other. Due to that, desynapsis is taking place, that is the pairing is also being separating from each other. And next one, what is it is been telling that during this stage there is not a complete separation is taking place, still the chromosomes, they are remaining attached to each other at a point in the form of X-shaped structure, and that is called chiasmata. Even though the dissolution of synaptonymal complex is taking place, thus there is not a complete separation taking place, and the chromosomes, they are remaining attached to each other in the form of an X-shaped structure, and that is called chiasmata. Okay, my dear children, and this chiasmata, number of chiasmata is depending on the length of the chromosome. And the last but not least related to the diplotin stage, it has been said that as desynapsis proceeds, the chiasmata starts moving towards end of the chromosome. So, chiasmata which is formed there in the center of the chromosome, it starts to move towards the end of the chromosome, and that process is called terminalization. So, it is related to diplotin stage. These 5 stages are very, very important, my dear children. The 5th one is diakinesis. Here the chromosome condenses further during the diakinesis, and this stage is known as moving through. And in this stage we can see the 4 parts of tetrad which is visible, and this stage homologous chromosomes separate further, chiasmata terminalize and making the chiasmata more visibly, or we can say more clearly. So, chiasmata which is formed there, it will be clear during diakinesis, and the homologous chromosomes, they are separating from each other during this stage. So, that is related to prophase 1, which is the longest phase, and these 5 sub stages are very, very important, my dear children. You have to understand about it. Next one is metaphase 1. Microtubules forms spindle fiber, and it is a second phase, and the tetrad from prophase 1, they will be lining up to the equatorial plate, and these tetrads, they are seen on the equatorial plate by the spindle formation taking place, where they are attached to the spindle formation. And next one is the anaphase 1. In this phase, the number of chromosome is reduced to half. So, anaphase 1 is an important stage in the meiosis 1, where the number of chromosome is reduced to half, and each pole receives one haploid set of chromosome. You can see here, it is during anaphase 1, chromosome which are paired there, they separate from each other and they will be moving towards the opposite pole of the cell and the sister chromatids, but they will be living together, or they will be staying together. Telophase 1, 2 haploid daughter cells are formed. Here 2 haploid daughter cells are formed, and these 2 haploid daughter cells, they are formed by the division of cytoplasm. During telophase 1, nuclear membrane and nucleolus reappear, spindle fiber disappear, and the cytokinesis divides cell into 2. So, after the end of meiosis 1, 2 haploid daughter cells are formed there during telophase 1. Now, this telophase is followed by cytokinesis, due to which 2 haploid daughter cells are formed, which are called dyad. And the stage between meiosis 1 and meiosis 2, that is called interkinesis, and this stage is short lived. So, after meiosis 1, meiosis 2 begins, but between these two stages there is a short period of interval, and that is called interkinesis. And after interkinesis, prophase 2 of meiosis 2 begins. So, we can see meiosis 2, it is similar to mitosis, or we can say it is also called equational division, and it consists of the following stages: prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. It is said that meiosis 2 is similar to mitosis. So, we can see in prophase 1 what is happening there: nucleoli and nuclear envelope disperse, and the chromatin reticulum, they will be shortening and thickening, and they are becoming rod like structure, which is called chromosome, and spindle fibers are forming. Next, in the case of metaphase 2, you can see here spindle fibers are formed from microtubules, and the chromosomes, they will be aligning themselves on the equatorial plate, or we can say on the metaphase plate. And anaphase 2, during this phase, the sister chromatids, they will be separated from each other and they will be moving towards the opposite pole of the cell. So, you can see the sister chromatids, they are separated from each other and they will be moving to the opposite pole of the cell. Telophase 2, nuclear membrane and nucleolus reappears, chromosome becomes long and thin, that is it is becoming chromatin reticulum. And spindle fiber disappears, and cytokinesis will be taking place after telophase. That is why after cytokinesis, what is happening there? Already 2 haploid daughter cells are formed in telophase 1 and 2 haploid daughter cells you can see here in telophase 2. So, together you can see 1, 2, 3, 4 haploid daughter cells are formed there after the cytokinesis. After the cytokinesis, 4 haploid daughter cells are formed there. 2 haploid daughter cells after telophase 2 and 2 haploid daughter cells are formed after telophase 1. In this manner, 4 haploid daughter cells are formed there. And what is cytokinesis? It is a division of cytoplasm, and as a result of cytokinesis, 4 haploid daughter cells which are called tetrad are formed. Okay, my dear children, that is related to cytokinesis. Now we can see the significance of meiosis or the importance of meiosis. It helps in the production of gametes. Number 2, the reduction in chromosome number is taking place because of meiosis. It helps to maintain the chromosome number in the somatic cells. Number 4, it helps in the production of variations which will be different from the parent cells, and the variations they are occurring in 2 ways, different combination of chromosome and by crossing over. I think you would have understood about the significance of meiosis. So, my dear children, already this topic you have studied in your previous class. It is just a brushing up session, and there are new technical terms related to the this chapter, such as synaptonymal complex, dyad, tetrad, bivalent, homologous chromosome, isn't it, terminalization, chiasmata. These things you have to understand and you have to study, because from this chapter question used to come in the question paper. You have to go through the figures also related to this chapter. It is very, very important. So, my dear children, with this chapter is completed. I think you would have understood this chapter. If you are having any doubt related to this chapter, you can call me and clarify your doubt. Please be on track and complete your notes. Don't keep it for the last moment, my dear children. Be always on the track. By saying this, I am winding the session here. We will be meeting in the next session. Till then, take care.